It's my pleasure to have today Dr. Aude Bernheim giving a talk in our seminar series. To be honest, I learned about the Dr. Aude Bernheim work in Twitter, because Twitter now is my main source of scientific papers. So in that Twitter, Aude volunteered to give a seminar, and I thought, okay, it would be great to have her at the institute and to explain her work. The end of the story is that she was extremely kind, and she accepted the invitation of the IBB. And well, it's my pleasure to give the word to Dr. Bernheim. So please let the questions for Aude in the chat, and at the end of the talk, well, she will answer the question. Okay, let's go.
So hi everyone, thank you for the invitation. I'm always super happy to be able to share my science. So I'm Aude Bernheim, and the work I'm going to present to you now is mostly what I did during my postdoc at the Weizmann Institute of Science in Israel, but I'm currently now based in Paris, France. So, and I'm going to discuss antiviral immunity in prokaryotes and eukaryotes. So, all cells, eukaryotic and prokaryotic, face the threat of viral infection. For bacteria, you face the threat of viral infection from viruses called bacteriophages, or phages for short, but also plants and, of course, humans. And so against these viruses, all of these cells, they have to find ways to defend themselves against these infections. And so when here I'm showing you an example of a bacteriophage, so the virus of a bacteria. So when it infects, it will be able to replicate and kill the cell. And so to prevent that, the bacteria will develop an immune system. And all the cells that I showed you will develop what we call immune systems or defense systems, preventing any infection. But in response to that, the virus will develop counter weapons and found ways to make the immune system irrelevant and be able to still infect. And this goes on and on and on. And so this arms race is critical in understanding that both hosts and viruses will be involved in this arms race, and this will drive to the diversification of immune systems. We will develop multiple types of weapons as a host that will be diverse. And this is one of the main explanations, this hypothesis, towards why we see very different types of immunity for different types of organisms. So for, I would say, the past 50 years first, the main defense systems of bacteria were called restriction modification enzymes. And then in the year 2000, it was discovered that bacteria also encode adaptive immune systems, which are CRISPR-Cas. But when we look into eukaryotic cells, the main immune systems, I would say, that are described are, for example, NLR and RNA in plants. And when we focus on a single, on a cell level, for example, animal cells, we will have the interferon response and the interferon stimulated genes, or ISTs. And so, for example, we don't find in plants or in humans CRISPR-Cas systems or restriction modification enzymes. And so what we believe is that this arms race is one of the reasons that we don't observe such type of similar and conserved immune systems across domains. However, this picture started to change just a couple years ago. And the main reason why this idea that is starting to shift is that discovering many more bacterial prokaryotic defense systems. So as I told you, for a while, we had restriction modification, and then we had CRISPR. There is also a cell type which is called abortive infection, which is quite specific and well described only in a few bacteria. It's supposed to be much more widespread, but has been described in specific cases. And I would say since 2018, we have now had a whole lot of novel type of defense systems, such as chemical defense, signaling systems. and a lot of families that we actually know they are defending against viruses, but we don't know how. And how is this possible? It's through a method that was developed by the lab I did my postdoc in, which allowed for the systematic discovery of microbial immune system. And the two examples I will dive in today really rely on this method of discovery. And it all started from an observation from Kira Makarova and Eugene Koonin that saw that in bacteria, immune systems cluster within defense islands. What does that mean? This means that when you take here, it's a line, and this represents each line is a genome, and each of these rectangles is a gene. When you look at one defense system, such as LMs or restriction modification, you see that next to it we have a CRISPR, or break system, which is also a restriction modification. So you see they're all clustered in defense island. And this is true for several types of genomes. So you see for this genome, we have CRISPR and RM, or ABI and restriction endonuclease. However, what we can also see is that there are genes that are in gray, and gray here represents unknown function. So the idea behind the method was to say if bacterial defense systems cluster together, maybe these genes in gray are also defense systems themselves. And this was an approach that was done and tested in that paper. And indeed, they were able to show that these genes in gray are defense systems. And they showed, like this, a discovery of more than 9 novel families of defense systems. And within this work, one of the systems that was discovered is the systems that had genes that had tier domains. And that was very exciting because tier domains are very important in the immune systems of both plants and humans. So this was kind of a first grasp at the idea that there are actual conservation between eukaryotic and prokaryotic immune systems. And further along, in 2019, another paper came out of our lab, which discovers that actually bacteria have a defense system that is an ancestor of the C-gas-Sting system, which is an important immune pathway in humans. And recently, through another method, it was also discovered that argonauts, which are a very important immune system in plants, are also encoded by bacteria. And so the question that that kind of drove my postdoc was, are there more out there? Are these just traces, or can we find other type of conservations between immune systems of prokaryotes and eukaryotes? And so I'm going to give you 2 examples. One I will dig deep into details, and another more lightly of example of conservations. One will be a lot of conservations at the conceptual but also molecular mechanism level, while the other is just to show how an inspiration from other forms of immunity, and notably eukaryotic immunity, can help us solve other problems. Okay. So the first story I want to share with you is about viperin, and I'll get to a second about what viperins are. And it was just published in Nature under this title, so please feel free to go check out the paper. So viperin is an immune protein from humans that has an antiviral activity, which means that when a virus comes in, the viperin the virus is sensed, the viperin is expressed, and we see antiviral activity. This was known for a while. However, it was absolutely not known how viperin work. And then in 2018, Gidi and colleagues were able to discover the molecular mechanism of viperins, which is absolutely amazing. So what is it? You take this modified nucleotide called CTP, so the modified nucleotide that is incorporated in the RNA, and the viperin takes out this water molecule here in red. And this creates here a double bond. And this creates this modified nucleotide called DDH-CTP. And this DDH-CTP is what is called an RNA chain terminator, meaning that when it is incorporated in an RNA chain, it is no longer possible to add another nucleotide because the 3' OH is missing. You cannot add following nucleotides because you cannot do the bond between the OH and the phosphate group. And so this mechanism, the production of this DDH-CTP, leads to pretty much this mechanism where the viperin is from CTP producing DDH CTP, which is incorporated in the RNA chain of the virus here, and then this gets prematurely terminated transcript, and this is the antiviral activity. So this paper came out in June 2018, and when we saw it, we were super excited because it was the first time that this type of immune mechanism was described, this type of RNA chain terminator being produced naturally inside cells to fight against viruses. And so we were just thinking, wow, it's really amazing. It would be so cool if bacteria would also do that. So we said, why not? And as I told you, we expected not to find anything because up to now, all the proofs and evidence showed that bacteria and eukaryotes don't have any immune systems in common, or very few. But of course, if I'm talking now, it's because we did find homologs of these human viperins in prokaryotic genomes. So again, here I'm showing you genomes with genes of bacteria this type, and these genes in red are actually homologs of the human viperin. And what did we find next to these genes? Well, here, everything that is in yellow are other type of defense systems, CRISPR, restriction modification. So for us, this was a sign that not only did we find homologous sequences, but also that the genetic context suggests that in prokaryotes, viperins also have an antiviral activity, meaning they would have the same function both in eukaryotes and prokaryotes. So then, in order to test this, what we did is taking one of these prokaryotic viperin, putting it heterogonously in E. coli behind an inducible promoter, and testing if this strain is able to resist phage. The graph I'm showing you now is the optical density through time. So this is a strain that doesn't have a pVip, it grows, and when you add here a virus, a bacteriophage, the population collapses. This is a sign that the phage infection was successful and the bacteria are dead. So now if we take our strains with the prokaryotic viperin, or they call them pVip, this is when you don't have a virus, it goes in a similar manner to the control. However, if now we add the bacteriophage, boom, it's able to still grow. And this is a sign that the strain is now resistant to the phage infection, which means that the pVip provided anti-phage activity. So that means that prokaryotic viperins, in a similar manner as the eukaryotic viperins, provide antiviral activity. So at this point, we wanted to have a bit of a better idea of what this family of prokaryotic viperin is. So what we did is that we took all the homologs we had, and one thing I want to underline is that it's only 218 pVips in around 60,000 genomes, which means that they are very rare. However, an interesting characteristic is that while being rare, you do see that they are in different clades with different phylum. So typically we have some pVips in Firmicutes, Archaea, Cyanobacteria, Protobacteria, Bacteroidetes. So very widespread. And so this kind of points out to a very ancient origin of this system. So one thing that we're curious about is where would the human viperin fall within this? And so when you look more precisely, what you see is that all the eukaryotic viperin are actually forming a monophyletic clade that stems from a clade of bacterial or prokaryotic viperin. And so this really points out toward the prokaryotic origin of the eukaryotic viperin. But one thing that is also super interesting is that we have this protein that is very important for the eukaryotic immune system, and it produces something important. However, because now we have these in bacteria, you can see that the eukaryotic diversity is really small compared to the prokaryotic diversity and all the prokaryotes that are encoding all the sequence diversity of the protein. But this sequence diversity could also mean that actually only some of the prokaryotic viperins are antiviral. Maybe other of these homologous sequence actually have another function. And so we ask, are all these pVips antiviral? So to do this, what we did was to take 60 homologs, so pretty much all over the phylogenetic tree. And then we tested them in the same manner that I just showed you. We tested them to see if they would provide antiviral activity. And what we see is that all these triangles here in red are viperins that provided antiphage activity. So 28 out of 60. For us, that was a lot. You could say it's only half. Actually, we expected much less. The reason is like we are testing the activity in E. coli. So it's absolutely not in their native condition. So of course, we expected some not to fold well, not to have the necessary host factor to function, etc. What we're very happy to see is that not only a lot of them did provide antiviral activity, but we had candidates from every single family that we could see were antiviral, really pointing out to a whole family of proteins being involved in antiviral activity across domains. And one kind of perks, because we are seeing that it was kind of working super well, for the fun of it, we actually tried to see if the human viperin would provide defense in bacteria against bacterial viruses, and actually it did. So the human viperin is here, and it did provide activity against prokaryotic viruses. And beyond being kind of cool to have a eukaryotic protein being able to defend against prokaryotic virus, for us it kind of pointed towards the idea that the mechanism would somehow be shared if this was also working in E. coli. And it would also provide us with the most fantastic positive control for the rest of the work. So at this point, we had all these very diverse proteins that would provide antiviral activity, so same function, but we have at the same time this diversity of sequence. And so we wondered if the sequence diversity would somehow reflect another form of diversity. And by this, I mean different molecules. We know the human viperin is producing a specific molecule. Do all these prokaryotic viperins produce the same molecule, or others, or something else? So the first clue to answer this question came from the observation of the, again, the genetic context of these viperins. So in humans, the human viperin, next to it sits a citidylate kinase, which is called CMPK2. And this kinase has a very important function regarding viperin. So the function is to take CMP, so the monophosphate version of the citidine, and to phosphorylate it into CTP, which generates then through the human viperin DDH-CTP. So pretty much the kinase is providing the substrate to the human viperin. And when we looked in our genomes, we found that for 20% of the viperin, we also had a kinase nearby. And in some cases, like in this bacterial genome, we indeed had a citidylate kinase. But for other bacterial genomes, we actually found, or prokaryotic genomes, we found other type of kinase, such as thymidylate kinase or adenylate kinase. And the way we interpreted this information is that if this is the reaction happening in with the human viperin, so the kinase going from CMP to CTP to DDH-CTP, maybe these are the reactions that are going on with the prokaryotic viperins, meaning that you have somehow GMP and then the kinase make GTP and then the prokaryotic viperins make DDH-CTP. Or UMP and then the kinase make UTP and then the pVips make DDH-UTP. So this was the first kind of clue that actually maybe pVips indeed would produce different type of molecules. So then the idea was to go and test for that. So how did we do that? We took prokaryotic viperin, we grow them, we pellet them, we take only what is inside the cells, and we only take the small molecule fractions, so we pass them through a filter, and then we analyze them through metabolomics techniques, so mass spec LC-MS techniques. And so I'm just going to walk through how it looks like. And for this, I will first take a look at what the human viperin looks like. So I told you that the human viperin, we know from the 2018 paper that the human viperin produces DDH-CTP. So this specific molecule. What we didn't know is that if we would be able to pick up a signal using this type of methods to pick up this type of nucleotides. And actually, we were able to, and the way this looks like is like this. This is on top the negative control, on the bottom the human viperin. And in LC-MS techniques, a molecule is defined in a unique manner by two types of information: the retention time on the column, and its mass. And so here we are plotting across retention time at a specific mass, the intensity. So when you don't see anything, it means the product is not there. But when you see a peak here, it means that this specific product, if it has a specific retention time and at a specific mass, it is there. So this is what the presence of a DDH-CTP looks like in this technique. However, when we looked a bit more deeply to this analysis, we not only found presence of DDH-CTP, we also found presence of DDHC and DDH-CMP, which is the same molecule, but with either one phosphate, so you remove two phosphate, or three phosphate without phosphate groups. And these are what they look like. And for us, it was quite interesting, because what happens is that the DDH-CTP is not stable in classical, like buffer conditions. It loses the phosphate. So what we see here is kind of residual molecules after the phosphates are gone, because this is not super stable. But instead of looking for just one molecule, we would look for kind of a spectra, a signature, derivatives of DDH-CTP, because these would only appear if you make this type of modification, which the viperins do. Okay. So using these type of techniques, what we did is we had the human viperin that we know produce DDH-CTP derivatives. And what I'm going to show you now is based on what I just described, but is a summary of the results. And so when we looked into our own pVips, we did find some pVip, pVip 6, pVip 50, that produce DDH-CTP derivatives. But we had several other pVips that did not produce DDH-CTP derivatives. However, given that we had the hypothesis with the kinases that they could produce other type of DDH nucleotides, we looked for other types of DDH nucleotide. And indeed, they produce DDH-UTP derivatives. And when we looked even more, we had some that produce DDH-GTP derivatives. And in all of these cases, you see that the viperins really produce one type of the nucleotides, but we actually discovered that we had a lot of them which were able to take either CTP or UTP as a substrate, and so to produce both. And even in some cases, for pVip 58, it is able to actually produce all the type of DDH nucleotides. And so this led us to conclude that actually the prokaryotic viperins produce diverse antiviral molecules. And very excitingly, the fact that they are able to catalyze the reactions from diverse substrate also opens the door for actually the catalysis of, for example, artificial nucleotides by just making this small modification that pretty much transforms the molecule into an RNA or into at least a chain terminator. One thing that I just want to mention, which I find very exciting, is that we also found that for 5 pVips, we were not able to detect any of these molecules. However, these 5 pVips provide a very clear antiviral activity, which means that these ones, we actually don't know how they work, and we don't know what they do. So it's possible that they produce a different type of nucleotide that we didn't pick up using our method. This is completely possible, or maybe through a different type of mechanisms. So at this stage, we know that the pVips produce diverse antiviral molecules, but we still needed to try to understand how they protect bacteria against phages. What was the mechanism? And so, as I told you, in bacteria, in humans, the way it works is that the viperin produces DDH-CTP, and this DDH-CTP is incorporated in the RNA of the virus. So what we hypothesized is that it was working the same way for prokaryotic viperin, that it would also be incorporated in the RNA of the virus. And so to test this, to test if pVips impact viral-dependent RNA transcription, we created a small genetic reporting system. So basically, we put a reporter gene, the GFP, under the control of a viral promoter. And in this case, the T7 promoter, because T7 is a phage that are sensitive to pVips. And so the way it works is that we induce the GFP, but only under the control of a viral polymerase. And if everything goes well, what we get are functional GFP transcripts, everything is okay. But now, if we add the prokaryotic viperins to the system, what we expect to happen is that those pVips will produce these DDH nucleotides, and that these DDH nucleotides will be incorporated inside the RNA of the transcripts. So really, and that these, once incorporated, this will generate prematurely terminated transcript, and then we end up with non-functional GFP, so no GFP signal. And one thing that is key in this experiment is that the GFP is under the control of a viral polymerase. The bacterial RNA polymerase is not sensitive, or at least doesn't seem sensitive to these DDH nucleotides. So you only repress specifically the transcription of here genes under the control of viral polymerase, so here the GFP. And so here is the type of graph you get. This is for a negative control. So in gray here, it's when we don't induce GFP, so no GFP. In green, it's when you induce only the GFP, and in red, it's when, so the positive control, it's when you induce the GFP and the pVip, and here in red, it's the negative control, so the curves are similar. And now for the human viperin, we saw that the GFP signal levels really were brought to basically the level of the negative control, so the signal is fully repressed, showing that it impacts properly the viral-dependent RNA transcription. And if we now do the same with a couple of our pVips, we see exactly the same type of phenotypes. We seem to show that the pVips impact viral-dependent RNA transcription. Just to be sure that it was really happening at the RNA level and not for example, the translational level, we just did the same experiment, but instead of looking at GFP, we just measure the level of GFP transcripts through RNA-seq, and we're indeed able to see that the transcription was reduced by more than 90% in these cases, showing so a very strong impact on the pVips on the viral-dependent RNA transcription. And so the model we currently have is that the pVip is able to take one or several nucleotides, generate these modified nucleotides called DDH nucleotides, and that then these are incorporated into the phage RNAs, leading to premature termination of the phage transcripts. So this is, as you see, models that are extremely similar to what's happening in the human cells. So really a conservation of pretty much sequences, mechanisms, and chemistry somehow. So, so one last thing that I kind of want to tell you on these viperins is that this is CTP, so a molecule I've been describing with you. But if we now look at this molecule, just replacing this group, which in the viperin is destroyed, you end up with AZT, which is a very famous antiviral against HIV. And actually, all these drugs, acyclovir, telbivudine, are also chain terminators. And somehow, it's interesting to see that from a therapeutical point of view, we kind of invented RNA or DNA chain termination as an antiviral in, I would say, an artificial way using these molecules. And it was discovered that humans, or human cells, do them by themselves to produce these type of antivirals molecules, and that actually bacteria invented them first. And so in another manner, what we can also think is that maybe all these new molecules, DDH-GTP, DDH-UTP, might also potentially become one day new antiviral drugs as RNA chain terminators. So just to conclude on this, so I just showed you the family of pVips, which produce diverse antiviral molecules, and this antiviral strategy is conserved between prokaryotes and eukaryotes. with a higher diversity in the prokaryotic world. And as I just explained, we could think that maybe these molecules could be one day adapted to serve in clinical use. But if you just take, again, a step further, you can think that this is one strategy of what is called chemical defense. But usually, when we discover that you have this type of strategy in prokaryotes, it's very likely that the chemical defense strategy is more widespread than just this specific family of prokaryotic viperin. It is likely that there are other types of chemical defense systems that exist. And so maybe, now that we know, and we do know that bacteria produce antiviral molecules, maybe we can treat them in a bit as a similar way as we did with antibiotics and go and try to hunt the antiviral molecules that would be produced by bacteria. And to study that, to really go within a framework of phage bacteria interactions to go and use bacteria as a repository for antiviral molecules. So that was the first story I wanted to share with you in details with really, as you can see, a very strong conservation at pretty much every level between these two systems, sequence, chemical mechanism. And the second story I want to share is not about bacteria and human, but more about bacteria and plants. And it's how really the comparative immunology helped us to solve a mechanism of novel prokaryotic immune systems we discover and kind of solve a long-standing mystery. And so the second story was done in collaboration with Adi Millman and Abigail Stocker-Avichai, who are two fantastic scientists I had the pleasure of working with. And the publication is called, it's about bacterial Retrons. This is the title. It's available on bioRxiv, but it will be published next week. So there again, you should have able to have access to the papers. So here again, I will start by the main term in the paper. Retrons. So what is a retron? So retrons are a weird type of nucleic acids that were discovered in the 80s. So it was discovered pretty much just by running a gel, and people saw that there was a weird small band at the end for some bacteria. This is how it was discovered. And they saw that it was a DNA, and by kind of a deeper study, they in this paper, they characterized what was this small DNA that was produced by Retrons. And so I'm skipping a lot of steps, but pretty much a Retron is composed of two parts, a Retron type reverse transcriptase, RT, and a non-coding RNA. And what happened is that you have the non-coding RNA, which will take a super specific structure like this. And with, for example, the two G's here, and then the reverse transcriptase will come and from this G start reverse transcribing and creating a complementary DNA, while the RNA part is being degraded. And so you end up with this RNA-DNA hybrid called msDNA. So this is a Retron. And Retrons have been studied, so since the 80s, biochemically in a very specific way. And they have been used as tools in synthetic biology, but despite all this, their function has remained elusive. We didn't know what they do. And the way we stepped in was kind of chance. So we were looking at the novel defense systems that we had discovered through this, again, this technique of defense island. And in our system, we had two genes, a reverse transcriptase and an ATPase domain. And here, what I'm showing you is a plaque assay. When here, no system here with the systems that I'm showing here. And here it's diverse phages. And pretty much this is serial dilutions of a phage on a bacterial load. So when you have big holes, it means you have phages that manage to infect very well. And you can see that here, in comparison, when you have the system, you are able to pretty much see that the phage cannot infect properly. So this is a phenotype of defense. So we have this novel defense system that seems quite strong against many phages. And when we inspected a bit more closely this defense system, we observed something that usually we don't see, which is that instead of being as a tight operon, there was always between these two genes an intergenic region. And this is something that is quite unusual and that we don't usually see. And actually, through some modification, both bioinformatics and experimental, we were able to show that this part is a Retron. This intergenic region encodes for a non-coding RNA with a specific structure, and this is a reverse transcriptase of a Retron. So the whole part encodes a Retron. And so we have a novel antiviral system that encodes a Retron. And through mutational studies, where we mutated pretty much either the reverse transcriptase or diverse very specific site of the non-coding RNA or the associated gene, we are able to show that the Retron properties are essential for defense. If you just mutate one gene in the non-coding region, you lose completely the defense. So the Retron properties are really essential. The other thing we are able to show with these mutational studies is it's not only the Retron properties that are essential, it's also the additional genes that comes with a Retrons. But so at this point, we thought, okay, maybe actually, because we had a novel system that encodes the Retrons, maybe all Retrons are involved in antiviral activity. And maybe this is the function of Retron. And so to test this, the first idea we had is that I described to you in the beginning that a Retron was composed of two things, a non-coding RNA and a reverse transcriptase. However, for our new system that encodes the Retron, we knew that there was this other gene, this ATPase gene, which we saw was essential for defense through mutational analysis. So we thought actually maybe all of these, the names that are here, are are known Retrons that have been studied experimentally and biochemically. And we thought maybe these also actually come within systems with additional genes, and they do not come by themselves. And when we looked a bit more closely, indeed, each of these genes came within additional genes to form systems. And so what we did was to take all of these operands, not just the Retron part, and we put them in E. coli and tested them against diverse phages. And what we were able to observe, so this one actually we didn't test, but all of the one we tested, we were able to see that at least from one Retron from each of these type was able to provide defense against phages, showing that basically Retrons do provide antiviral activity against diverse phages. I'm not going to go into details here. What I just want to underline, these are all different types of Retrons, and then here you have different types of phage that you see that, and red means defense. You see that Retrons can protect against diverse phage, even if they are from the same type. So it's quite interesting to see that you have one type of system, but that is able to protect against very diverse viruses. So now that we knew that Retrons were defense systems, we wanted to know how they work. So here I'm going to skip quite a lot of the experimental work. You can find it in the paper. But pretty much what we discovered is that Retrons function through abortive infection. What does that mean? Abortive infection, so here we have the Retron and the editor, it means that when a phage infects this cell, so this cell is infected, the Retron will be activated, and the cell, the fact that you have the Retron, it will lead you to kill yourself. And because you kill yourself, the phage cannot replicate, and so you end up with a bacterial colony that survived. So it's kind of an altruistic suicide. And this is the abortive infection phenotype. So that's what we were able to show with the Retrons, meaning that the Retrons lead to the cell performing growth arrest, and we're able to show that on all the Retron systems we tested. And so knowing that pretty much it leads to cell death, we decided to try to understand what triggers Retrons. So to study this, what we did was to do what escapee strategy. So I showed you this type of graph before. So you have the phage concentration. Here you see that pretty much at the end of the serial dilution, you only have like 2 plaques, so 2 phages. This is when you have the defense system, the specific Retron EC48, and you see that it pretty much defends really well. But if you look a bit more closely, you have here actually a small plaque, and this phage was able to replicate. And so it's able to replicate even if the cell is encoding this defense system. And usually what happens is that this phage is a mutant. It has a mutation compared to all the other phages that allows the phage to overcome the defense system. So we took that phage, we sequenced it, and actually we did this on several things, and we looked into what gene we found mutations. And for 2 viruses, we did this. And interestingly enough, the mutations were in 2 different genes. But the 2 genes, which are very different, had the same function. They inhibit RegBCD. Okay, so what is RegBCD and what does it have to do with phage? So RegBCD is a complex that is important for DNA repair. However, it's basically an exonuclease. However, it's also a complex that will pretty much degrade a DNA of the phage when it comes in. And so when the phage comes in, RegBCD degrades the phage DNA, so you can kind of think of it also as somehow an immune system. And so you degrade this, and so in order to prevent that, what do the phages do? They encode these RegBCD inhibitors, and that allows to inhibit RegBCD and to get a successful infection. And so what we found is that we had phages that to be able to actually successfully infect in presence of Retron, they have to get rid of this RegBCD inhibitor, which means that somehow the inhibition of RegBCD triggers the system. And this is when we kind of, this made us think of a quite famous strategy in plant, which is called the guard hypothesis. So in plant, it has been shown on multiple occasions that one way the immune system is able to monitor the state of a cell is not by specifically recognizing a protein of the virus, but actually by recognizing something that has been made modified on a host protein. So typically you have a host protein, the pathogen comes, it will modify it, and this specific modification will be sensed by what is called a guard protein. And this guard protein, once it's sensed that the host protein has been modified, will lead to an immune response. And so for us, it really looked like this was exactly what we had under our hand. The host protein is RegBCD. When the phage comes, it inhibits RegBCD, so this is modified. And then the Retron, which is here the guard proteins, is able to sense the modification. And what was very interesting with this specific hypothesis is that in the plant immunity world, they had done several types of experiments that allowed to confirm this hypothesis. And so we just took this experiment and adapted them in bacteria. And so this actually led us to be able to conclude, I'm skipping the experimental part, but this led us to conclude to this model where basically you have this EC48 Retron that guards RegBCD. And then when the phage comes in, it expresses RegBCD inhibitors. RegBCD is modified, and so the Retron senses this RegBCD inhibition, and then it's able to activate the effector part, which then leads to cell death or growth arrest. Okay, so this is the model we currently have about how these specific Retrons, and this was really cool because it was the first time we were able to see that we had this type of guardian proteins in bacteria. So really a novel type of immune strategy that was again also present in bacteria. And one thing that I have to say about this is that it's quite interesting because it's pretty much this strategy is dangerous. It leads to your death. So it is dangerous. So you have to be careful not to trigger your own death in a very easily manner. And here you can think of this strategy as a second line of defense. RegBCD is somehow a first line of defense, meaning that it's able to cut the phage DNA. But when it's compromised, because the phage has compromised this line of defense, then the system, the suicide system, goes into action. So you only kill yourself when you basically know that you are almost already dead. So, and that's, I would say, kind of very complex and interesting strategy implemented by bacteria. And so to conclude on this part, it was very cool to be able to kind of crack this 30-year-old mystery of the Retrons physiological function, to discover guardian systems in bacteria, and to also see this evolutionary convergence in entire antiviral immunity, that you get this strategy somehow of evolutionary convergence into guarding a host protein instead of doing something else. And just to conclude on the overall thing, I hope that these two stories kind of convinced you of the power of what I call comparative immunology. One thing that we know that because we see that some systems are conserved across domains, is that maybe for some species, like here I'm showing you Emiliana huxley, which is a eukaryotic bug that is super important for the oceans and for which viral infection has tremendous implications. And the immune systems of Emiliana huxley are not known. But maybe now we know these immune systems are conserved, maybe we can try and look for specific things within this type of organisms. There is also concepts from eukaryotic immunity. I told you that from a eukaryotic immunity point of view, for example, we applied the Guardian hypothesis and discovered one example in bacteria, but maybe we know a lot more about eukaryotic immunity than bacterial immunity. So we could use this concept to discover more things about prokaryotic systems. So here I put interferons, so maybe one day we'll discover that bacteria also have something quite similar as interferon, meaning some form of quorum sensing system specific for viral infection, maybe one day. What we can also do is the other way around, because we have this conservation, use bacteria as model to study immune mechanism. I showed you, for example, that for the viperins, the mechanism for human and bacteria are very similar. And you have outstanding questions, such as how do you do self versus non-self recognition within these systems, meaning how are the host polymerase able to function with these nucleotides, but not the viral polymerase? And these questions, you could get answers from bacteria where it's much easier to work, and then apply them to, for example, humans. And finally, one thing that I'm always super excited with for working with prokaryotes is that you have this fantastic diversity. And I think in the viperin example, it really shows that when you have one thing cool that exists in humans, if by any chance you find it in prokaryotes, then you are blessed with the fact that they will encode tons of versions. And this has already been extremely useful for all the innovation that stems out of the field of phage-bacteria interaction, such as restriction modification system or CRISPR-Cas, where you have the same principle, which is super useful, but then all the flavors really allow you to go for very diverse application and kind of tailoring what you want to do. So to conclude, I just want to say pretty much thanks to everyone who was involved in the project. So this is a lab. So this is my postdoc advisor, Rotem Sorek, who was absolutely instrumental and amazing postdoc advisor in this discovery. And then for the Retron, as I say, all the work was done with Adi Milman and Abigail Stocker-Avihail, really. Three of us equal, and then other help. And from the viperin, really also again Adi Milman, who I love working with, and a lot of people who helped me both from the experiments and the design, let's say, and also people from two different companies, MS Tomics for the mass spectrometry and Panton for some biochemistry, EMBO for funding, and the Weizmann Institute in general. And finally, I just want to tell you that this is where I am now. I moved back to Paris. I'm working for INSERM, and these are type of questions that that I'm interested in. And so this is just to give you a vibe of what I hope I will do in the next coming years. Thank you.
Thank you all. Always on talk, so I don't know if someone has questions. I have many. So there is one question from Thomas. So he's asking, can you read or do you?
Yes, I can read. These viperins can be stimulated or used as treatment for RNA viruses. What kind of problems can appear if it is possible to stimulate it? So I think I was just reading a preprint this morning that shows that apparently, for example, if we are talking about SARS-CoV-2, it was shown that it seems some of the RNA viruses play with the interferon response. So first, a disclaimer, I'm not a human immunologist at all, so I know very little about human immunology. But what I read that it plays with the interferon, and viperin is one of the genes that is the mostly expressed after interferon induction. What people are actually looking at, it's less to stimulate it than to use the molecules produced by viperins against these viruses. And it seems from, again, not my publication, but there is a preprint that showed that in vitro, apparently, the DDH-CTP, so only the one produced by the human viperin for now, they didn't test the others because we just published the paper, so they didn't know they existed. They tested the DDH-CTP, and they saw a very strong repression of the SARS-CoV-2 polymerase. So my guess is that they are trying it on more on other systems. I'm actually starting a collaboration with virologists to test all these molecules on diverse RNA viruses and diverse cell lines. So I hope I'll have an answer at some point. We don't know. And so I think, yeah, for now, the idea is more to use these viperins, the product of the viperin, than to stimulate the viperins themselves.
Okay, thank you. There are a couple of questions, so are mine questions. So the first one is if some of the species of bacteria that you characterize with VIPs, of course, are present in the vertebrate intestinal microbiota, and if you think that can have a role in defending of virus in the maybe intestinal virus or.
Yeah, so I think that in general the ecological role of viperin will be very hard to assess, and I have two things on that. So first, as I said, they're very rare. So I would guess that most bacteria would defend themselves against viruses through other means. This is not like the thing that, just to give you a comparison, 90% of bacteria have RM systems. Okay, so it's like this, the way you would defend yourself is first restriction modification. Then you have a lot of other defense systems, but like 218 out of 60,000 genomes, that's like very, very rare. A second thing is that here I only focused about the characterization of these pVips, but in a lot of cases, they are not alone. They come within systems. So they come with 2 or 3 other genes. Actually, I can show you, I have a slide on this. I will share my screen again. Maybe this up. So I don't think I shared the correct thing. So here are examples of viperins within systems. So you see you have the pVip, and in a lot of systems, this is the most common configuration. Next to it, you have an ankyrin repeat gene and ICLE RNAs. The ankyrin repeat is a bit, it's known to be involved in recognition. It's something that deals with protein-protein interactions. So we think that maybe this is the part that kind of senses the virus, and that then it activates the pVip. Okay. And then maybe the like RNAs, and it degrades the rest of the RNA, something like that. We don't know. But you see that it can also come in, sometimes we find it by itself, the pVip. And then we have this form or this form, which I think tells us that we don't know exactly if what it does in these cases is the production of this DDHP, or maybe it modifies another form of nucleotide. Maybe it's only activated upon certain occasion. They are still like it's really the beginning. So I think we don't really know yet exactly how it works, but it's possible that it works into the intestine, and if it does, in the like the virus and the microbiome are super important. So what are the implications? I don't really know, but yeah, it could be involved in that.
Okay, thank you. And the other question that I was wondering if the structure of these pVips are homologous also human or the vertebrate or the animal pVips? Because you mentioned or you talk about the active domain, that it's the SAM domain, but some authors think that the N-terminal and the C-terminal domain also has some kind of antiviral activity. So I was wondering how they look like.
So currently we don't have any structures of the prokaryotic viperins because they just been published, sadly. I really hope some will come out soon, but I have several things to say about this. So first, there was very nice papers on eukaryotic viperins. And to be honest, I'm quite sure some structure will come out soon because we know there are other people working on these pVips that were kind of working on stuff that I think are similar, but more from a biochemical point of view. So these teams, there are teams that are working on the structures of viperins, and they also started looking at other viperins, so eukaryotic, fungi, these kind of things. And they do see that you do have a observation. It's also interesting because it comes from a protein family of radical sum enzyme, and it has these families as other members, such as MOA, which is a GTP cyclase. And it's from actually the structure, the fact that the GTP cyclase of MOA and the viperin looked very similar, that the hypothesis about the fact that it's an enzyme that has a substrate that is triphosphate came around. And so what they saw when they did the structure is that you had a lot of similarities, and in these studies they were trying to understand where do the human viperin substrate specificity comes from. Why does it take only C? And they saw that there were a few amino acids that were very important, and they found actually also another enzyme, another viperin, that was able to be a bit less specific, but they didn't have yet all the pVips. So when these studies came out, I started looking with my sequences and trying to map them on a structure. And what I can say is I'm quite sure there are other things that are important because the different amino acids underlying are not enough to explain the type of substrate specificity we are observing. As for another thing on the N-terminal, C-terminal, so one thing is that indeed in bacteria we don't have we remove, for example, for the human viperin, it has a plate that tagged into the ER, and this we removed. But however, even while we removed this, it was still very specific to C. So this doesn't seem to be something, but maybe something else is there. And the last thing I wanted to say about your question, the last answer, is that even though we do think that, for example, for phage T7, this is a mechanism of the pVip, for other phages, we do see antiviral activity, but for example, with some pVip, I said we don't see these DDUH nucleotides. And so maybe you have other types of antiviral activity, and this is also an hypothesis for the human viperin, that you have another type of activity. So I'm quite convinced that we will still discover other things within the antiviral mechanism, and that maybe, for example, some pVips are able to take very modified nucleotides, and within the cycle of phages, you do have these modifications. So I'm quite convinced that we'll discover new stuff.
Thank you, Aude. It's been an interesting talk.
Thank you.
If there are not more questions, so we can close the meeting. And well, there is no. Okay.
Thank you.
Thank you very much.
Thanks, Aude.
Thanks. Bye.
Bye.
Have a nice day. Bye bye.
Thank you.